Yes, yes, absolutely.
Thank you. So a very healthy and beautiful evening to one and all virtually present here. I would like to extend my warm wishes on behalf of the joint collaboration of INYAS and INYAS. Today, I welcome you all to the fifth lecture of the lecture series under Teach for Science program. So dear participants, it gives me immense pleasure to introduce Dr. Nishima, one of the distinguished researchers in the field of nanoscience. She is currently working as an assistant professor at University Institute of Engineering and Technology, Punjab University, Chandigarh. After the completion of PhD in chemistry from Institute of Microbial Technology, CSIR, and Punjab University, Chandigarh in 2010, she joined as a postdoctoral fellow at School of Material Science and Engineering and School of Biological Sciences, Nanyang Technological University, Singapore, jointly with Technion, Israel from 2010 to 2011. In 2012, she was appointed as a project scientist at National Agri-Food Biotechnology Institute, DBT Mohali, and later was awarded a DST inspired faculty position at Center for Nanoscience and Nanotechnology, Punjab University, Chandigarh. Her research interests are biosensing of contaminants using nanomaterial and peptides, development of antimicrobial conjugates of peptides and nanoparticles to combat multi-drug resistance, targeted drug delivery using nanoparticles, self-assembly of nanomaterials. She has published numerous publications in the reputed international journals, along with 2 patents. She has supervised several PhD, master's, and undergraduate students, along with 1 postdoc. In her academic career, she has added several prestigious awards to her name. Presently, Dr. Nishima is a member of multiple professional bodies, namely Wise Women in Science and Indian Academy of Sciences, INYAS. Also, she is a reviewer of several journals related to nanoscience and has been awarded 9 projects. So now I request our distinguished faculty, Dr. Nishima, to kindly deliver her talk on the advancement of biochemistry and drug delivery. We welcome you, ma'am.
Thank you so much, Rajesh. That was a very nice introduction. Thank you very much. I don't know whether I deserve so much of admiration or not. I would be more happy if the students present here would be able to understand the things that I'm going to talk about. So before I start, I'll just like to share my screen.
Yes, ma'am, please.
Okay, so welcome you all. The topic of my talk today is advancements in biosensing and drug delivery. So please don't get scared of the topic, which states that these are advancements, because the advancements which I'm going to talk about in these 2 areas, I'll try to make them as simple as possible, because I can understand that the audience here is primarily from BSc and MSc. So in between, if you are not able to understand any particular topic or any particular word, please stop me. I would like to explain it then and there so that we can move to the next slide. So you are free to have any kind of interaction that you want to have. I would not like to be like a one-way process where only I am speaking. So it would be better if we can have an interaction sort of a webinar. So even I'll be asking you some things, which so you have to be very, very attentive. So advancements in biosensing and drug delivery. So these are the 2 main things that I'm going to talk about today, which is biosensing and drug delivery. Now, why, first of all, I've chosen this topic. The reason why I have chosen this topic, especially for an audience like you, is because, as you know, in the past couple of years, we have seen this COVID pandemic, and we know the importance of drug delivery. And in fact, we also now know the importance of diagnostics as well. So I'll be touching slightly on that topic also. Sensing again is a very, very important part, especially when it comes to medical things. For example, if you want to detect some kind of a disease, you should know, you should have the infrastructure, or the simplest of the simple infrastructure to sense that particular analyte. Now, when I use the word analyte, it will mean that something which you want to detect. Okay, that is an analyte, something which you want to detect. Now let's go to our first slide, where you can see these three pictures on your screen: biosensor, drug delivery. So what I have done is I have taken this picture from a very good journal, RSC Advances. If you can see, these two circles are overlapping: biosensing and drug delivery. I in the coming few slides, you will understand the meaning of this actually. In the other slide, in the other diagram on the left hand side, you can see what is this. This is actually the characteristics of a biosensor. Okay, I will not go into the deep details of what comprises the biosensor, but in very, very simple terms. For example, you have an analyte. You see the third diagram. You want to detect that analyte. Say, for example, you want to detect glucose. Okay, simple. Glucose you want to detect. You want to see if the person is suffering from diabetes or not. So if a person has diabetes, obviously he or she will have increased glucose level. So let's see if we can detect glucose. So you have glucose, any kind of other analyte, you have a matrix, then you have a transducer, which will change the signal. Then you have an amplifier, which will amplify the signal, and finally a display unit, which will display your sensing. So basically something which uses a biological element for sensing a particular analyte, that is a biosensor. Now this I'm telling in very, very simple terms. Now, biosensor kaisa hona chahiye? What should be the characteristic of the biosensor? बायोसेंसर फर्स्ट ऑफ ऑल उसमें ये जरूर होना चाहिए कि इट शुड बी स्पेसिफिक. स्पेसिफिक का मतलब क्या है? अगर आपको ग्लूकोज की बायोसेंसिंग करनी है, सो दैट पर्टिकुलर बायोसेंसर शुड ओनली डिटेक्ट ग्लूकोज एंड नॉट अदर थिंग्स. मे बी इफ समथिंग एल्स इज प्रेजेंट, से फॉर एग्जांपल एनी अदर थिंग इन द सीरम इज प्रेजेंट, इट शुड नॉट डिटेक्ट that thing. It should only detect glucose. Next is sensitivity. Sensitivity, it should be sensitive enough to detect very small concentrations of that particular analyte. Of course, it should be biocompatible. Biocompatible ka matlab kya hai? Whenever you use some material, it should be compatible with your body or any living thing that you are using. So that should be biocompatible. And what now this whole thing जो भी मैंने आपसे बात की, this was mainly about biosensing का concept. What is biosensing? Now when you detect that particular disease कि okay, हमने sensing से पता कर लिया कि जो there is some problem, there is some disease, and now that disease needs to be of course cured. तो वो curing के लिए means जो therapeutics के लिए हम drug I mean the medicine, it has to be delivered at a particular site. Let's say take the example of cancer. So in cancer, you have to deliver the drug at that particular site. Say for example, if it is a stomach cancer, you want the drug to only go to the stomach and not to any other organ. Okay, so you will design some kind of a mechanism so that there is a proper smart drug, targeted drug delivery. So the drug will reach only that site. तो वो हम कैसे करेंगे? अब advancements इसमें क्या है, which is the title of this talk, advancements in these two fields. So advancements ये है, when we bring in nanotechnology into biosensing and drug delivery, immediately these are advancements. Why? Because जो problems हमें कन्वेंशनल मेथोडोलॉजी से आती हैं इन बायोसेंसिंग एंड इन ड्रग डिलीवरी, वो प्रॉब्लम्स को हम ओवरकम कर सकते हैं विद द हेल्प ऑफ नैनो साइंस एंड नैनो टेक्नोलॉजी. राइट, आई एम वेरी श्योर कि आपने पिछले लेक्चर्स में भी और वैसे भी नैनो साइंस नैनो टेक्नोलॉजी के बारे में सुना होगा, पढ़ा होगा, नैनो पार्टिकल्स के बारे में भी पढ़ा होगा. आई डोंट नो, राजेश, इज इट ओके इफ वी इंटरैक्ट विद द स्टूडेंट्स और शुड वी टेक अप एट द एंड?
Ma'am, it's up to you.
Okay, okay, fine. So I would like to then ask if anyone out of you would like to tell me what is a nanoparticle? Maybe you can raise your hands. So if there's no answer, then I will assume that you don't know the meaning of a nanoparticle. Then I will go into the explanation. If anybody, okay, I'll read the chat, I think. Yes. So someone has answered, Muskan Thakur has answered that nanoparticles are the particles which have size in nanorange. Very good, Muskan. So I will just like to correct it a little bit. So at least one dimension should be in the nano size range. Okay, so at least one dimension. कुछ चीजें होती है जो centimeters में भी होती है, but उनका say for example width is in the nanometer size. So very good. So these are nanoparticles. So these build up the nano size, nanotechnology domain. Somebody else has also answered. Small particle ranges between 1 to 100 nanometer of size. Great. So we can go ahead now that I know that you know the meaning. Okay, but say you see there are so many participants, 29 participants over here. Out of the 29 or let's say 27 participants, only 2 have answered, right? So it is safe to conclude here also that nano size is not widely understood, though it is prevalent everywhere, but it is not very widely understood. You can see from the screen, I have found out from this data from website, link is given below. So 24% of Americans have reported to be heard a lot or some about nanotechnology, right? And 44% of Americans report having heard nothing at all about nanotechnology. Now, why I'm specifically highlighting on this American study, because अगर US में ही awareness इतनी कम है, so what can we say about our own country where we have many other problems? So awareness regarding this is still much lower than what it should be. But at the same time, it is this education is very important. Nano education is very important because it has applications in numerous areas, not only biosensing and drug delivery, but numerous other areas. And apart from that, for you students, I want to tell that one of the advantages is that studying nano is really fun. It is really funny to go into this world of nanoscience. जो बिल्कुल different है bulk world से. The properties are completely different and very interesting properties. And one thing which I like the most about this field is that it breaks down the disciplinary boundaries. Now, what are these disciplinary boundaries? Disciplinary boundaries means that, say, I would say I am from the chemistry field. Someone is from physics, other person is from biology. So we tend to categorize science into different fields. He's the chemist, he's a physicist, biologist, so on and so forth. So nano size में क्या होता है कि you have to study about chemistry. आपको physics भी पढ़ना है, आपको biology भी पढ़ना है applications के लिए. You need to know basics of computer science as well. So nano education breaks down all these disciplinary boundaries, and this is one of the best thing about this nanoscience. अभी कुछ स्लाइड्स, 1-2 स्लाइड्स में और मैं आपको थोड़ा सा इंट्रोडक्शन और थोड़ा सा इंटरेस्टिंग पार्ट बताऊंगी इस फील्ड के बारे में. देन वील गो स्ट्रेट इनटू द एक्सपेरिमेंटल पार्ट. Okay, now I ask you to do another thing. Please read this sentence. आप अपने यहाँ पढ़िए, ये सेंटेंस पढ़िए. Okay, I hope you have read it by now. So in the time it takes to read this sentence, your fingernails will have grown approximately 1 nanometer. So I found this very interesting. That is why I have shared this with you, is that how small, usually we say how small is a nanometer, but I would like to say from here you can see how big is a nanometer. Big not in terms of, I'm right now saying in terms of size, but big I'm saying in terms of how interesting or how special is this size domain. You see, by the time we have read this sentence, 3 times we have read this, so your fingernails have grown how much? 3 nanometers. Again, so if you have ever been to a football match, you know how big a football field is. So if you want to cover, they say, imagine this, that you have to cover the football field with a 1 nanometer thick layer of paint. And for that, how much paint you will need? The answer is that you will just need 1 teaspoonful of paint. That's it. Okay, so this is how big or how small, it's a relative term, whatever you want to call it, how big is a nanometer or how small is a nanometer. Okay, so my next question, and you can see the answer also in the screen. Did scientists actually create nano? Did scientists create nano? क्या scientists ने nano चीजें जो हैं वो create की थी, या फिर these were, or the other sentence is correct? No, it was already there in nature. If you think it was already there in nature, can you write me an example in the chat box? How do you think it was already there in the nature? Or have you seen or learned or heard about any example of something, uh, some natural thing which is nano in size? DNA. Bacteria, I would say no. Yes, DNA. Okay, great. So scientists actually did not create DNA. We can say in other terms that it is God who is the inventor of these nano things and everything else. So good that you know that it was already present. Okay. So this is just a clear depiction of, if you go from this is the wing of a butterfly. If you see from centimeters, we're going to micrometers, then this is how it looks like in micrometers. And then if you go to the nanometer domain. Now, why it is so important? Why studying nanoscience, and why it has so many applications, particularly in these two areas, which I'm going to discuss today. This is because when a thing is made small, it leads to new properties. And out of these new properties, the most important, just सबसे important property है nanomaterials की, that is their surface area. Surface area nano-sized particles का बहुत high होता है. Now, why this is so? Why the nano-sized particles have a high surface area? Again, if you can answer me this question in the chat box, it will be good. Otherwise, in the next slide, I'm going to give you a sort of a mathematical numerical. Can you answer how surface area of nano-sized particles is higher than the, let's say, bulk particles? Just try to correlate it with the radius, with the diameter, and think about this. Think about the formula of surface area, maybe of a sphere, of a circle. If you know what is the formula for the surface area. Okay, I have received now one answer. So again, Muskan, it is due to its smaller size, surface and volume, surface to volume ratio. I think what you mean is surface to volume ratio increases. Yes, surface to volume ratio increases, definitely increases. Now let's see this. So how does it increase? Ab carefully dekho. The rest of the people other than Muskan. See here, this is a single particle. It has a radius of 1 centimeter. If you can see the blue ball on the left hand side, right side pe dekho, 8 particles of radius 0.5 centimeter, right? Both of these have the same total volume, 4.2 centimeter cube. Got it? So for a fixed total volume, decreasing the radius by a factor of 2 doubles the surface. कैसे किया double surface? Now let's go, इसमें जाओ ये जो squares. So the surface area of this blue sphere will be 12.6 centimeters square, while the surface area of these 8 particles of radius 0.5 centimeter. Okay, we have decreased the radius by a factor of 2, remember. Surface area has increased to 25.1 centimeter square. So we can safely say crushing a 1 centimeter particle into nanoparticle increases the surface area thousands of times. Okay? So this is just an example. This is a simple calculation to show how the surface area increases. This is why nanomaterials are so much widely used in as catalysts also. आपको पता है ये catalyst क्या होते हैं? Catalyst में surface area कितना important है? So this is how these are used. Smallness leads to new properties. Reactivities increase, change in melting point strength, conductivity, color. So you see, this is bulk gold. ऊपर जो bulk gold है, you must have seen bulk gold definitely. but gold nanoparticles, they don't look at all like the bulk gold. These are red or purple or blue in color. Okay, this is another example that stained glass. Stained glass is a glass which was being used since centuries, and it contains a mixture of gold and silver nanoparticles. I will not go into the details of the phenomena which leads to this kind of color in these nanoparticles. Basically, it is surface plasmon resonance. In case anybody is interested, you please write down in the chat box. I will take it up. Otherwise, I will stick to the very basics. So as you change, the very interesting property with these nanoparticles is कि जब आप उसका size change करते हैं, so when you change the size, there is a change in the color, or when you change the shape, there is a change in the color. So the properties are different when their sizes are different. आप बल्क मटेरियल में ऐसा नहीं होगा, बल्क मटेरियल में गोल्ड जैसा भी ले लो वो गोल्ड ही रहेगा, ठीक है, इट्स प्रॉपर्टीज विल रिमेन द सेम. बट व्हेन यू गो इनटू द नैनो डोमेन, द प्रॉपर्टीज दे कंप्लीटली चेंज. And then we have nano silver products. You very well know antibacterial. These are very much there in everybody's homes. Okay, another question to you is, can you name any product which you have seen, anything you must have seen, any advertisement on your televisions of some antibacterial nano products? Water filters. Mukul Mahajan, right? Water filters. Anything else that you have seen? Maybe in medicines or some electric equipments. Clothes, paints. Very good, very good. Now I can see other students also answering. That's very nice. Great. So you know that a lot of things now contain the safe nanoparticles. Maybe you have used sunscreen also. Have you ever tried to read what are the constituents, what do you say, ingredients in that sunscreen? Try to see them. If you have some small kid at home, these drops are given, what they call vitamin D drops. Just check what is there in the formulation. Okay. So lot many nano-sized things are actually now available. They are available in the market as well. So especially these antibacterial products, they are used highly in the wash. I think Samsung is one company. Yes, Samsung is the one which uses these nano silvers a lot. Girls must be knowing what is this. And in toys also, in the covers of the keyboards, antibacterial keyboards, etc., socks, nano sunblock, which I was just telling you. So again, I would like to stress upon it that this field is a total interdisciplinary endeavor. It requires contribution from various kinds of fields, and it has applications in various areas: semiconducting, electronics, medicine, defense. There's no field which is without the help of nanoscience. Now let's come to the technical part of this presentation. Since I formed the basis, what is the introductory part, let's come to the technical part. So the main thing which in my research area, which I'm focusing on in my lab, is we are trying to address the healthcare area. So in that, amongst the drug delivery area, we are targeting cancer. Right now, we are targeting cancer. We are also targeting microbial diseases, especially the strains which are multi-drug resistant. Food-borne diseases. Food-borne diseases are very common, especially in our country. In India, food-borne diseases are very common. And in this monsoon seasons, you must be hearing a lot of food-borne disease cases are being reported. And pesticide-based problem. So this is one area that I'm going to talk about for the biosensing thing, where we have detected pesticides. Then again for drug delivery, epilepsy, and Alzheimer's disease. So as I have just described to you what a nanoparticle, they have small size, large surface to volume ratio. Third thing, this is a little dicey thing. I really don't like to add this third point, that these are easily synthesized and easily functionalized. They are not easily synthesized and easily functionalized. It takes a lot of effort to synthesize these nanoparticles. It seems that it's a very easy synthesis, but it is not. They are highly prone to aggregation, and then their purification is also sometimes very tedious. But since their applications are very good, since the biocompatibility is very good, so it is worth the synthesis. So this all actually started with we were thinking of developing some strategies for synthesis of these nanoparticles for biosensing, but using a green synthetic route, because we wanted to make biocompatible nanoparticles. So we studied the literature and found that the reducing agents, so basically when you are synthesizing, for example, gold nanoparticles, these have zero oxidation state. So in gold chloride, which is the salt that is used, तो उसमें क्या होगा, gold chloride को आपको gold zero में convert करना है. Do you know what is the oxidation state of gold in gold chloride? In tetrachloroauric acid, HAuCl4. Now, can you tell me what would be the oxidation state of gold here? HAuCl4. Yes, Anusuya. Yes, right, Muskan. So it is +3. So from +3, we have to go to 0. So what we have to use is, we have to use a reducing agent, because we have to reduce it from +3 to 0. So normally the reducing agents that were being used were very toxic. So we switched to amino acids. These are naturally occurring amino acids. And we found that we could easily synthesized good monodispersed nanoparticles, gold nanoparticles. You can see there, these are the red bars. The red bars shows the concentrations versus the kind of amino acids that we have used for the synthesis of these nanoparticles. And then, of course, we have to do the characterization. So the peak at 520 confirmed the synthesis. Now, this is a specific peak for some particular size of nanoparticles. So if there's a peak shift, accordingly, we can understand that there's a change in the size of the particles. And the width of this UV absorption peak refers to the dispersity of these nanoparticles. If you have a narrow dispersion, you will have a narrow UV peak, the absorbance peak. Okay. Then we can use an equipment called the dynamic light scattering equipment, which can give you the size dispersity, कि कितना size, कितने number of particles, कितने size के हैं, ठीक? And the image below is that of TEM. Here you can see that these particles are very, very nice and monodispersed and of the size of around 10 nanometers, right? Okay, so then later on, we also studied how we can synthesize different shapes of this. So now, not only we were able to change the size of the nanoparticles, but we were also able to tune the shapes of these nanoparticles by variation in the reaction conditions. So this also we studied thoroughly. And based on the success of gold nanoparticles, we decided to try it with silver nanoparticles as well. And yes, there were some challenges with silver nanoparticles. It was not as good as that of gold, but yes, particles were being synthesized. And the important thing was, कि जो silver nanoparticles हैं, these are antimicrobial in comparison to gold nanoparticles, which are not antimicrobial. In fact, we found out that जो histidine वाले particles है, D image में जो है, these proved to be very good antimicrobial agents. तो हमने Department of Microbiology के साथ collaboration में इसकी कुछ और studies की, and we found out that it was acting as a very good antimicrobial agent, और wound healing पे, burn wound models पे ये बहुत अच्छे से act करता था. So what we did now, we drew into picture the peptides. Peptides क्या होते हैं? Do you know what are peptides? Peptides are sort of polymers of what? Basic unit क्या होता है peptides का? Yes, amino acids. Amino acids are the basic units, right, Anchal and Mukul and Muskan? So these are the, so amino acids, they combine. Why is it called amino acid? Because amino means amine group, acid means the carboxylic acid group. So they join through amide bonds, okay, and form the peptides. So there are some particular peptides which are very interesting. I don't know whether you have heard about them or not sometime. These are called cell penetrating peptides. ये देखिए सामने लिखा है ना, cell penetrating peptide. Cell penetrating peptides कैसे peptides होते हैं? These are usually positively charged peptides which are not very long and they're able to penetrate into the membranes. तो इसमें क्या usefulness है कि जो आप इस पेप्टाइड के साथ जो भी कोई कार्गो लगाएंगे, इट विल बी एबल टू टेक दैट कार्गो अलोंग. ठीक है, तो अगर आपको ड्रग डिलीवरी करानी है, यू हैव टू टेक द ड्रग इनटू द पर्टिकुलर membrane which is sort of impermeable. You attach the drug to these peptides and the peptides will carry it to the site. Okay, so what we did was we took the nanoparticle, जो हमने nanoparticle synthesize कर रहे थे, and then loaded the peptide onto it and the drug onto it. Drug means जो भी हमने, say for example vancomycin, we are using, it is an antibiotic, so you have vancomycin, you have the CPP. CPP is cell penetrating peptide here, and you have the gold particles. Now you target the bacteria. Now otherwise the loading of vancomycin, जो आपको कितनी concentration चाहिए थी vancomycin की for killing particular amount of bacteria was very high. But when we use this kind of methodology, the amount of vancomycin used was very less as compared to that of without the CPP and the gold nanoparticles. So this was the advantage of using this system. So here again, we synthesized these silver nanoparticles and studied these. Left side, you can see the TEM images. Right side, you can actually see this is a, these are SEM images of Klebsiella pneumoniae. Okay. So if you can see this A part is the intact cell of this bacteria, Klebsiella pneumoniae. Okay. And I think C is the undisrupted. Yeah. A prime is the one which is treated with HAG. HAG means the histidine capped silver nanoparticles after 60 minutes. So you can see the disruption of the bacteria. You can clearly, visibly see the destruction. Bacteria ki destruction start ho gayi hai. So, and here you can see in the C, you can see the totally disruptive ends of this bacteria. So it proves that this system is acting very well against these kind of bacteria. Okay, then yeah, then we also studied the penetration of CPP conjugated gold nanoparticles, and similar results we were seeing in these gold nanoparticles as well. But in case of silver nanoparticles, we also found that there was synergistic activity, which means there was a combination of the activity of silver plus the antibiotic. So these are confocal micros. These are microscopic images, आप समझिए, but you can see these clearly with dyes. कुछ dyes डालते हैं, ठीक है, जिससे कि there's uptake of the dye and you can clearly see whatever the things you want to see. So here we have seen the uptake of CPP gold nanoparticles, and with it is labeled with a dye called FITC. This is in gram positive and gram negative both, E. coli and Staph aureus, and we can see that there is killing of the cells. So as I said, that Department of Microbiology, they tested these particles and they found that these were really very effective against the Klebsiella pneumonia induced burn wound infection. So it was good for skin infections. In fact, we also carried out the cellular studies to check if it was biocompatible and or not, because it is very important कि जो हम बना रहे हैं nano system, it should be biocompatible, because if you go down to very low sizes of nanoparticles, say 2-3 nanometers or 1 or 2 nanometers, then they tend to be toxic. So you have to keep that thing also in mind. So how they are used for theranostics or for drug delivery, especially in the case of cancer. Now I'm going to focus on cancer for the next 2-3 slides. So what are the therapies used in treatment of cancer? I'm sure आपने ये तीनों नाम सुना होगा, surgery, chemotherapy, radiotherapy. You must have heard of किसी ना किसी के बारे में तो cancer से related कोई बात सुनी होगी. So what do you think is the problem? Problem in the sense कि cancer तो problem है ही है. Cancer की treatment में what are your observations? What are the problems? जब हम treatment करते हैं cancer patients का, so what are the problems that the cancer patients they face? Please write down in the chat box. Any example of the problem that they face, and what do you think is the reason behind that problem? One is the disease itself, but the other thing is that you are giving a treatment. You, for example, you're giving chemotherapy. Now the chemotherapy does absolutely, Archana, very good. So the therapy affects the normal cells also. This is what Archana has written, side effects. Yes. So because the drugs do not know, ठीक है, अब drug body में है, drug means medicine. अब उस ड्रग को तो नहीं पता ना कि ये कैंसर सेल है और ये नॉर्मल सेल है. So it will kill the healthy cells as well, right? So this is the problem. Yes, it affects the healthy cell. Now, how you can overcome this problem? Any idea how you can overcome this problem? You write it down in the chat box, how you can overcome this problem. Any idea? Till the time I will discuss something about surgery. So surgery is one thing, but surgery kab applicable hogi? Surgery will be applicable only when it has not affected many areas. It is targeted onto a single area, cancer. Then only surgery is good. Otherwise, there's no point of surgery. So I had asked you what is the solution? Cell-targeted therapy, targeted delivery, targeted drug delivery. Yes. If it is specific to these cells, right? If it is specific to, so specific कैसे बन सकता है? स्पेसिफिक ऐसे बन सकता है कि जो कैंसर सेल्स पे जो आपके रिसेप्टर्स हैं उनके अगेंस्ट आप स्पेसिफिक मॉलिक्यूल्स जनरेट करिए, उन मॉलिक्यूल्स का एक ड्रग डिलीवरी सिस्टम बनाइए जो कि उसको straight away it will go and bind to that specific receptor. So that is how it is termed as a targeted drug delivery, right? So it goes into that particular targeted site only. Yes, by developing specific biomarkers. Good. In case you find it too basic, just let me know, because I'm happy to see good response from you in the chat box. So I think some of the terminologies you already know, though I don't know which field you are from, BSc, MSc, which exactly biology or chemistry. That is why I'm using a mix of both. So limitations of the currently used therapies are they have numerous side effects. This you already know. You must have seen those effects also. Poor efficacy, systemic toxicity, MDR for some drugs. And then there is relapse also. More than 90% cases, there is relapse after a certain period of time. So can nanotechnology provide a solution to these problems? Can you answer me in a yes or no? If you answer yes, then it would mean that you have already seen some example. So can you type in the chat box, what is the answer to this? I have received 1 yes, 2 yes, 3 yes. So 3 people are saying that yes, it will provide a solution to these drug delivery problems. Okay, so unfortunately, not many people are saying that it will provide a solution. Okay, so let us now, I have to convince you how it can provide solutions. So this is one solution. So since we have already published it, so I can guarantee you that this is one solution which can be used if it is properly extrapolated. Okay, so here what we did was that we synthesized and delivered the size-tunable cationic peptide functioning old nanohybrids in the cancer cell. So here we use a truncated TAD peptides, a specific peptide sequence TAD peptide. And we use that peptide itself for the synthesis of the nanoparticles. And then we loaded doxorubicin. Doxorubicin is an anti-cancer drug. And we checked whether it is able to penetrate first of all into the cancer cells, and then it is whether or not it is able to kill those cells. So let's go to the other side. So this is the whole schematic of this drug delivery system. You see this yellow color, these are the gold salt. This is the gold salt, and then we reduce it and then pegylate it. Pegylation is done to increase its circulation into the bodies, and so that the body does not recognize it as something as a foreign material and does not generate antibodies against it. Okay, so this is how we generated this thing, and we use the passive targeting method. right now here we did not use an active targeting, means the ones that we have just discussed. अभी हमने बात की थी ना, targeted delivery पे attach specific biomarkers and all that. So that is an active targeting. For cancers, you can also use passive targeting. Passive targeting में क्या होता है, because cancer cells, they have a leaky vasculature. So there's a effect called the enhanced permeation and retention effect. उसकी वजह से they have a leaky vasculature. So the drugs, they or the compounds, they effectively can be internalized. And on top of that, we have, remember, we have a cell penetrating peptide. Okay, this is the major thing. So we synthesized at different pH, we checked what is the size dispersity of these gold nanoparticles and how we can use these. So you see these are the actual cellular images taken by a fluorescence microscope. And here are the bare gold nanoparticles. You can also see different, we've used different dyes, lysotracker, FITC, DAPI. DAPI is a dye which stains specifically the nucleus, okay? So we can see that these are able to internalize, this whole nano construct that we had synthesized is able to penetrate into the cells, and hence the loading of the drug can now be very less. तो अगर जो हमारी एंटी कैंसर ड्रग है उसकी लोडिंग जो मतलब उसकी जो कॉन्सेंट्रेशन है हमने कम कर दी है, ठीक है, उसकी जो रिक्वायरमेंट है वो कम कर दी, तो अगर हमने एंटी कैंसर ड्रग की रिक्वायरमेंट कम कर दी है, इट डायरेक्टली पॉइंट्स आउट दैट द साइड इफेक्ट्स विल बी सिग्निफिकेंटली रिड्यूस्ड. Okay, now on top of that, if you have an active drug delivery system, it will again significantly reduce the side effects. So these are definitely the solutions to these problems with drug delivery. ये जो एक image है, this is the confocal Z-stack. Z-stack means that you have, suppose, let's say you imagine the cell as a potato and you slice it like this, and then you see, visualize all these slices. So this is what this means, the Z-stacking. So the advantages of the formulation that are based on targeted delivery over the conventional anti-cancer therapy means are the selective targeting and enhanced delivery of the drugs, reduction in the dose and of expensive. Sorry, I forgot to tell you that these drugs are very, very expensive. Now, because a common man cannot afford अगर एक medicine मतलब एक what do you call, one strip is of 2,000 rupees. How can a poor man afford such expensive drugs? So it is very important that the dosage, if the dosage is removed, the cost of the drugs will also be reduced. Increased solubility, yes. The normally the anti-cancer drugs are water insoluble drugs, so there's a big problem of solubility. Reduction exposure of drug to other organs, and phototherapy, photothermal property of GNP means gold nanoparticles. They can be used. What is photothermal therapy? If I don't know whether I should go into the details, but I will quickly tell you what is photothermal property. Gold nanoparticles जब उस proper cancer area पे bind करेंगे, then if you irradiate with IR radiation, NIR, then it will take up the heat and it will kill the cancer cells. So healthy cells जो हैं वो intact रहेंगे, cancer cells will be killed. Yes, somebody has also answered in the chat box, passing light and emit thermal to treat the cancer cells. Yes, very good. Okay, so these we had some discussion about cancer therapy. एक छोटा सा काम हमने किया था where we tried to develop formulation. आप में से कुछ लोगों ने देखा होगा कि जिन लोगों को एस्थमा होता है, they use a kind of inhaler जिसको वो inhale करते, उसमें drug होते, it inhales and they can breathe easily. So we tried to have such sort of a thing, inhaler sort of a thing, where we can have the anti-epileptic compound there for the delivery of this to the brain. What is the problem? What is the problem when something it has to be delivered to the brain? There's a huge barrier. What is that barrier? Do you know what is that barrier which has to be crossed for anything that wants to go to the brain? What is that term that is used? Okay, so there's no answer. It's called the blood-brain barrier. Blood-brain barrier. So it has got, so it is difficult to cross the blood-brain barrier and reach the brain. Just like if you, yes, B. Just like if you have some boring lecture and you are not able, the lecture is not able to go in your brains. So just imagine a boring lecture is equal to this blood-brain barrier. Now you will never forget it. So we had used bioengineered PLGA chitosan nanoparticles for brain-targeted intranasal delivery. Now, the tricky thing that we did was we used intranasal method of delivery of this anti-epileptic drug. Intranasal delivery हमने क्यों? Intranasal means through the nasal route, nose के through. So we used TRH and thyrotropin releasing hormones. So this is a drug which is used for this epilepsy, anti-epileptic. So we use this, and here you can see this is the synthesized anti-epileptic drug, and we encapsulate it into the PLGA nanoparticles. You can see these polymer. This is a polymer, PLGA polymer, and we loaded the anti-epileptic drug into this. This drug is also a peptide basically. This TRH analog is also a peptide, and this PLGA के polymer में encapsulate कर लिया, and then we did a chitosan coating onto it. Chitosan coating हमने क्यों की? Because this chitosan has mucoadhesive properties. Mucoadhesive means it can bind to the nasal membrane, so it will be easy for us to administer it into the brain. So we checked in mice models, and we took the brain tissue, did the analysis, again used nanoparticles. So we had a polymer-based nanoencapsulation system, and the detection was also based on the nanoparticles. So we did see that, yes, there was good amount of drug loading, and the biocompatibility was there, the drug release and the internalization into the brain tissue using the nasal delivery route was actually successful. So for the biosensing part, we did the pesticide detection, right? So these are the nano conjugates that we had used. Just a minute. This. So what is a pesticide? Pesticide is a chemical or a biological agent that deters, incapacitates, kills or otherwise discourages pests. So अब थोड़ी देर के लिए आप diseases से pesticides पे आ जाओ. Why this is important? Detection of pesticides हमारी country में we know we are especially in this northern India, lot of we depend a lot upon agriculture. तो agriculture की output को increase करने के लिए we have to use pesticides in order to kill the pests, but sometimes we overdo that. We overuse the pesticides. Overuse से क्या होता है कि these enter into the food chain and then they magnify, biomagnification हो जाता है, right? So it causes problems. So these are the disasters, some examples of the disasters. Particularly, I'm very these two things, the last two. You see the incident in Bhopal, and then this Bihar school meal poisoning incident 2013 में जो हुआ था, इसमें 23 students they died in the school. वो क्यों हुआ था? क्योंकि जो मिड डे मील में जो उनको खाना मिल रहा था, the oil that was used, it was stored in a container which was infected with the pesticide, organophosphorus pesticide. So this was the reason that this was one thing which had struck me the most at that time, and therefore we started with this work actually at that time, long back in 2012 already we had started. Also heavy metals, they cause a lot of contamination and disasters. And what are the conventional methods? वैसे कैसे हम pesticides को detect कर सकते हैं? हम HPLC use कर सकते हैं, GC use कर सकते हैं, we can use mass, immunoassays. But the problem, what are the problem with these kind of equipments? First of all, these are very high cost equipments. I'm sure आपने ये use कर रहे होंगे आप अपनी graduation, post graduation में. Then you need to have skilled personnel to use them, and you need to have sophisticated lab setup for them. But abhi kya need of present time kya, that the detection methodology should be simple, cheap, and sort of an on-site detection method should be there. कि जहाँ पे problem है जिस site पे, वहीं पे detection हो पाए. You don't need to bring the sample. अब आप GC को इतना बड़ा या HPLC को उठा के तो नहीं ले जा सकते ना site पे. You have to bring the sample over there, and you need to purchase those equipment. So maintenance is there. So when I got this Inspire Faculty Award of Cold Nanoparticle Based Detection of Pesticide in Food and Water in 2012, I started this work. So do you see this? So you see this picture I saw somewhere at that time is of she's a girl called Bakhtiar Kaur. She has cancer, and her you see the graying of her hair. So this cancer belt you must have heard about in Punjab. So there's a lot of problem of pesticide, indiscriminate use of pesticides, especially in this Punjab region. So what can be there? We have some kind of a simple sensor where you don't need to use such sophisticated equipments, and you don't need expert personnel. So we tried. So first of all, we chose ethyl parathenic organophosphate pesticide, and we checked कि अगर हमने जो gold nanoparticles बनाए हैं using cysteine, जो मैंने आपको पहले बताया था amino acids वाले, so can we use them for detection? We saw that yes, we can do. इसमें क्या है कि एक एसिटाइल कोलिनेस्टरेज एंजाइम होता है. तो अगर तो इथाइल पैराथाइन प्रेजेंट है, मतलब ऑर्गेनोफॉस्फेट पेस्टिसाइड प्रेजेंट है, सो इट ब्लॉक्स द एक्शन ऑफ दिस एसिटाइल कोलिनेस्टरेज, एक जो एंजाइम है. तो ये जो कन्वर्जन है एसिटाइल थायोकोलिन का, वो थायोकोलिन में नहीं हो पाता, राइट. एंड इफ दिस थिंग द pesticide is not present, then this conversion can occur. Okay, so based on this we could get a yes no kind of a sensor कि हाँ इसमें present है, हाँ इसमें नहीं present है. और कितना present है और कितना नहीं present है वो गोल्ड के कलर से हम बता पा रहे थे. So कितना गोल्ड एग्रीगेट हो रहा था वो डिपेंड कर रहा था कि कितना पेस्टिसाइड उस सैंपल में प्रेजेंट है. So we found out that we could go to a detection limit of 0.081 nanogram per ml, which was very good, depending seeing that we were using such simple methodology. Then we checked FORATE also. For FORATE, we did a simple method where we used aptamers. Aptamers क्या है? जैसे नॉर्मली हम लोग एंटीबॉडीज यूज करते हैं. सो एंटीबॉडीज आर जनरेटेड इन एनिमल्स, तो उसमें एनिमल वर्क चाहिए होता है. एप्टामर्स क्या है? दीज आर स्मॉल ओलिगोन्यूक्लियोटाइड्स, डीएनए और आरएनए सीक्वेंसेस. तो ये केमिकली आप सिंथेसाइज कर सकते हो. सो नो एनिमल वर्क इज रिक्वायर्ड फॉर दिस काइंड ऑफ एप्टामर सिंथेसिस. Then we detected pesticide called malathion in the environment. Sorry, this image has got disturbed a bit. तो इसमें हमने एक पेप्टाइड केटाइनिक पेप्टाइड यूज किया था, and there was certain kind of methodology that we used for the sensing. So when malathion was present in the sample, the gold nanoparticles got aggregated, तो उनका color change होता है from red to blue. और कितने एक्सटेंट तक वो कलर चेंज होता है, that is proportional to the concentration of the malathion. So this is how we were able to detect the presence and absence of that analyte, and also the concentration of that analyte. We also extrapolated this methodology to the detection of toxic metal ions like lead using these plasmonic gold nanoparticles, and we detected up to a limit of 37 picomolar. So now what we are doing is we are generating a sort of an automated assembly in collaboration with IIT Chennai. We are developing this methodology where we can detect this malathion using a simple device. So that device is already almost done. So very soon we'll be bringing it over into the market. So apart from the nanoparticles, metal nanoparticles, we have also used semiconducting nanoparticles. These are called quantum dots. ये 10 nanometer के नीचे ही होते हैं. The good thing about them is that they are very good. They are very good fluorescence. तो उनकी size अगर 1 nanometer size भी change होता है, accordingly their fluorescence also changes. तो हमने इससे detection की थी SEB की, Staphylococcus enterotoxin B. It is a toxin. So detection of toxins can also be done using nanoparticles, namely quantum dots. इसमें ज़्यादा details में नहीं जाऊंगी because of scarcity of time. So, and we are also utilizing self-assembled material. There is probably some disturbance. I have muted. self-assembled materials for drug delivery. You know that multi-drug resistance जो है, multi-drug resistance मतलब कि the bacterial strains they have become resistant. ठीक है, overuse of medicines over the time, तो bacteria या virus जो भी हैं, they develop methodologies to evade that जो इफेक्ट है उस एंटीबायोटिक का, राइट. सो ये चीज तो बहुत इंपॉर्टेंट हो गई है गिवन द सर्कमस्टांसेस ऑफ कोविड एंड नाउ न्यू डिजीजेस आर कमिंग अप. So we have tried to use these amino acids in the self assembled form, जो अपने आप self assemble हो जाते हैं. Normally भी ये process हमारी body में चलता रहता है, right? For example, जो diseases होती है Alzheimer's, Parkinson's, this happens because of amyloid generation, which means that there is aggregation of these amino acids. So but हमने इस चीज को use किया है, we have used this kind of aggregation, this kind of self assembly. इसको हम किसी useful चीज के लिए देखना चाहते हैं. So this assembly, we have used a diethioflavin T. तो इससे हम देख सकते हैं कि कितना वो टॉक्सिक है, वो जो एमाइलॉइड्स है. बट नाउ व्हाट वी आर डूइंग इज हम इसको यूज कर रहे हैं एज एंटी माइक्रोबियल ट्रैप्स, right? So we have found that some of these self-assembled amino acids have proven to be sufficiently antimicrobial. This work is under progress. Patent has also been filed. And these, you can see the beautiful SEM micrographs of the self-assembled amino acids and how these change in the concentration of the amino acid, change in the type of the amino acids, it changes the morphology of the resulting self-assembled nanostructures. And these are, some of them are toxic, some of them are not toxic, depending on the concentrations. So especially we have found that the glutamic acid self-assembled ones have proven to be very good antimicrobial in nature. And this paper, this we have already recently published, tyrosine silver nanocomposites. This is very good, I think a very good piece of work, because this can be directly used in the market as antimicrobial coatings, especially in food packaging. And these are the other applications of these antimicrobial constructs. So now coming to the summary, which the things that I've talked to you is I started from the green nanotechnological approach, and we went with major healthcare issues like the drug delivery in the case of cancer, epilepsy, and then we saw how a heavy metal ion or toxin detection, the pesticide detection, the biosensing can be done with the help of nanoparticles, and how with a combination of the nanoscience and with the help of biological science like the peptide science and a basic knowledge of chemistry can be used for applications in the field of drug delivery and biosensing. So this resulted in various research projects, patents, and journals. And this is the business model that we are now developing, like I just discussed with you. And lastly, I would like to acknowledge my department of Applied Sciences UIT for giving me the opportunity to do this kind of a work. Department of Chemistry also for the active collaboration, INYAS for giving the platform to disperse this knowledge to the youngsters as well, and to the various institutes where I did my PhD, postdoc, and the collaborators, and the various funding agencies which have given the funding to do this work. And I would like to just end with this thing that though I have given you solutions, the science never solves a problem without creating 10 more. Nanoscience definitely has given a solution to various problems, but then if it is misused, just like any other science, there can be some other 100 other problems also. So we have to be very careful. So this is where I would like to end. Thank you very much.
Thank you so much, ma'am. It was very informative talk, and it was amazing to learn about advancement of biochemistry and drug delivery. So allow me to open this talk for a Q&A session. And meanwhile, I am just dropping down thank you, the feedback form in the chat box. So students, participants, if you like to ask any question, you can open your mic and ask to the speaker directly. Or otherwise, you can, if you are feeling shy or having a technical problem, you can also put your question in the chat box.
If you're having a technical problem, then it's okay, but otherwise, please don't feel shy. Just go
Yes.
in and ask whatever is there in your mind. Rajesh, these are from BSc, which field?
Uh,
Chemistry, biology? I think these are from biology.
yeah, I think they're mix.
Mix.
Yatin,
Okay.
Yatin knows better actually.
Okay, no problem. Chemistry too. Somebody is writing chemistry too. Good. Good. So if there is any question in your mind, you can just write down in the chat box. Although we had an interactive session, you were answering my questions, and I'm very happy about it that you were answering the questions.
May I?
Yes, please go ahead.
Yes, please.
Yeah. Thank you for that session. Actually, I just want to know about that. You mentioned about that we are using these nanoparticles for this pesticide and I think malathion quenching and all that. So I just want to know that if we are releasing these nanoparticles in the environment, open environment, not in a closed environment, then do we have some long-term adverse effect of these nanoparticles on the environment? And do we need to quench it, these nanoparticles as well out of the ecosystem? So
Yeah, I got your point. Thank you. I think it's a very, very important question. I just slightly touched upon it when during my presentation when I said that, and in the last slide also, I just mentioned the same thing. It is very important to keep into your minds about the toxicity of the nanoparticles, because as I said, when you go into the nano range, the size changes, the properties changes, surface area enhances. So what happens is when you go to the very small size, like for example, 1 to 5 nanometer size particles, they are usually very toxic. But the particle size that we are dealing with is the particle size from 30 to 45 nanometers, which are not toxic. Again, the toxicity also depends upon the type of ligands that are there on the nanoparticles, because the ligands are those which will directly come into first that they will interact with the surroundings. So that is why the main purpose of our study was, first of all, to find out green synthetic routes for the synthesis of these nanoparticles. Only then we went on to use their applications. Again, the third thing is that for the concentration that you're using. So for example, even if there's some kind of a nutrient, if it is in a particular concentration range, then it will act as a nutrient. But if you go beyond the particular range, if you go into the excess range, then that nutrient can also be toxic. Okay, so it is not an absolute term, but I think it's a relative term. You need to first check what is the size, what is the shape. Shape is also another important thing. Usually spherical nanoparticles are not as toxic as the ones with the higher aspect ratios. And then the ligands, which are the ligands on top of the nanoparticles. So that is also very important to be taken care of. I'm glad that this question came up. I hope I have
yeah.
answered your question correctly.
Yeah. Thank you so much. Because I have read about these particles. It's like a double-edged sword. So we are
Yes.
just going it with the flow, but we don't know the exact any adverse effect, because most of the scientists are believing that if there is any type of side effect, then it will be a long term and after
Yes.
going through these experimentations. So thank you so much, and thanks for that enlightening talk. Thank you so much.
Thank you. I think that's a very good term. It is in fact a double-edged sword. So we have to be very careful.
Yeah, thank you, ma'am. There is a question from Mukul Mahajan in the chat box. Like, can you tell us more about quantum dots?
Yeah. So quantum dots, I didn't go deep into that because that's a completely somewhat different area. I think those who are from physics would be able to understand it clearly. So when you go into this quantum dot zone, which means there is quantum confinement, which means that if you understand the energy levels have now become discrete. So when the energy levels have become discrete, and you have a particle size in this range of 1 to 10 nanometer, which means below 10 nanometer, then the quantum confinement occurs. So these are called as quantum dots. For example, the quantum dots that we have synthesized are cadmium telluride quantum dots. These are highly fluorescent quantum dots, and the dots that we have synthesized are water soluble. So the thing is that when you tune the size, when you're synthesizing these nanoparticles in the same set of experiment, we get the green colored, starting from the blue to the green to the yellow and then orange and then red emit. In the same experiment, we get these different colored nanoparticle quantum dots. So they have different fluorescent emission. So they have a wide range of absorption, and they have very nice emission peaks. So instead of the organic dyes, you can use these quantum dots, which are highly fluorescent, highly emitting, and they have very less photobleaching also. So these are the important things about quantum dots that I think you should know.
Thank you, ma'am, for the answer. There is another question from Dr. Kuljit Kaur. She is requesting, like, can you elaborate more about heavy metal detection by nanoparticles?
Sure, why not? So this is one thing that we are really working upon. I did not include many slides on this. We are working on arsenic, mercury, and lead. Lead we have already published. Arsenic and mercury also we are doing. So why this? Of course, you know why we are doing the heavy metal detection. It is very important to determine the heavy metal contamination, especially in soil and water samples in and around the Punjab region. A lot of contamination is there in the samples, because then this enters, as I told you, it enters into the food chain, and then it magnifies, and then it goes into very high levels. So the detection scheme that we are using, I will just tell you very in basic sense. So we are using very specific aptamers against these metals. So we have aptamers against cadmium, arsenic, mercury. So when we use these aptamers, the important thing is that these will specifically bind to these particular ions, because they have a very good binding affinity to these ions. And then we design a system like we have designed with the gold nanoparticles. We have also done with the help of quantum dots also. In fact, with the quantum dots, we are having a very good detection ability. So we design the aptamers. We have a system where when the aptamers are loaded and when these metal ions are present. So when this whole conjugate, it recognizes the metal ion, it binds to it. Why? Because aptamer is there on our nanoparticle, and then it is specific to this metal ion, so it goes and binds to it, and you get a signal. When you get a signal, you analyze that signal, and you know how much concentration is there depending upon a correlation graph. So that is how we do it. If you want to know more details upon it, I can send you the papers. You can go through it. I think that will really help you a lot.
Thank you so much, ma'am, for the answer.
Sorry, there's a direct message also. So I just want to answer. Somebody has asked how to reach to you for this kind of research. So you can reach to me on my email ID, which I had given. It is nishima@pu.ac.in. Any kind of research paper that you want to know, or any kind of research-based knowledge that you want to have, if I can give it to you, I'll be more than happy. Thank you.
Yeah, thank you, ma'am. Thank you for sharing your email with students. So once again, I would like to request, if any of the participants have any questions, you can turn on your mic or you can use the chat box. Okay, so let's move towards the conclusion. If we do not have any more questions, on the behalf of organizing committee, once again, I thank you very much, Dr. Nishima, for honoring us with your presence. And we are looking forward to seeing you in person sometime in the near future. And once again, I would like to extend my sincere thanks to the participant for their presence in this lecture at Teach for Science event organized by Prayas with INYAS. And see you next time. Take care.
Thank you. Thank you.
Thank you.
Bye.